Hi, I'm Carlos Moreno, Chief Scientific Officer of ResonanceDx, and I'm excited to be here at the Georgia BioSummit Rapid Fire Showcase to talk to you about our technology in rapid point-of-care diagnostics. The first test that we're working on at ResonanceDx deals with a very significant health problem, and that is that of septic shock. Septic shock is a major cause of mortality in the US and also around the world. 30 million patients are affected around the world each year, and 200,000 Americans die of it each year. It is the third leading cause of death in the US and is a major cost and stressor for hospital systems. Recently, a new drug was FDA approved, known as angiotensin II, for the treatment of septic shock. It is currently a third-line vasopressor and hasn't shown a lot of uptake because it's more expensive than other vasopressors. And when you look at all patients, it doesn't show much more benefit than the current vasopressors. However, a recent study has shown that in a substantial subset of patients who have high levels of the biomarker serum renin, these patients have a 20% reduction in their mortality if they get angiotensin II. So the key is to identify those patients who will benefit from this drug. Unfortunately, current tests for angiotensin II take days to get the results, and patients need results in minutes, or physicians do, if they're going to make a decision on how to treat a patient and for that patient to benefit from this drug. So ResonanceDx has developed a renin test that gives results in less than 5 minutes. It is very simple to use, doesn't require any sample preparation or reagents, and it's also up to an order of magnitude more sensitive than current ELISA-based testing. It's also a flexible platform technology that can really be used for any test that's based on antibodies. The technology is based on bulk acoustic resonance sensing, or BARS, and it is based off of silicon wafer piezoelectric resonators that have on their detectors covalent links to antibodies of interest. When a biofluid flows over those resonators, then antigens can bind to those antibodies. Now, when the antibodies are excited on the piezoelectric resonators, they have a natural resonant frequency. They vibrate sort of like a wine glass if you run your finger around the rim and you get a tone. And if you add wine to the wine glass, that changes the tone. The same is true here. So if the antigen binds to the antibody, there's a change in the mass and stiffness of the antibody, and that changes the natural resonant frequency, which can be detected instantaneously. So this shift in resonant frequency corresponds to a positive result. In addition, these chips have multiple resonators on them, allowing us to multiplex the tests and include positive and negative control antibodies for calibration. Shown here is the layout of our chips, which shows 6 resonators, as well as a calibration for the electronics. And the technology, the chip is then wire bonded to a printed circuit board, which is part of a single-use disposable test cassette that allows for biofluids to flow through the fluidics into a chamber and over the devices. The test cassettes are inserted into a handheld instrument, which then automatically calibrates, runs the tests, and provides the result to the user in a setting such as an emergency room or intensive care unit. The fact that these are silicon wafer-based technology means that it allows us for rapid, inexpensive, and scalable manufacturing, which can keep the costs of our tests quite low, potentially on the order of $10 to $12 for a single-use disposable test cartridge. In addition, our results are very rapid, so we can get saturation in less than 10 minutes and provide results in less than 5 minutes. So this is very exciting and important for a point-of-care test. Shown here, you can see that we can actually detect very good signal-to-noise ratio and multiple different resonant peaks in the 1 to 3 gigahertz range. We can also see here that we get a shift after binding of renin to our detection antibodies that is in the megahertz range and easily detectable through our software analysis. Also shown here is the sensitivity of our devices, which compare here looking at detection of SARS-CoV-2 spike protein in saliva, comparable to PCR testing. So highly sensitive, far more sensitive than most other antibody antigen-based testing for SARS-CoV-2, and all with a 3-minute incubation. Our plans for bringing this to market include going with a FDA 510(k) predicate, using the laboratory renin test as our predicate and showing equivalence of our tests to the standard tests. In addition to our septic shock intervention panel that we're currently working on, we're also looking at other tests that we could potentially follow that include an at-home test for chronic kidney disease, discriminating bacteria from viral infections in primary care settings to reduce overuse of antibiotics, and discriminating COVID-19 from influenza, potentially, so that you can determine if someone should get a Merck drug for COVID-19 or Tamiflu. We have a potential strategic partner already in that La Jolla Pharmaceuticals manufactures angiotensin II, and our test could potentially greatly increase the sales of angiotensin II and bring it to a standard of care for the patients who have high levels of serum renin. The total addressable market for this first test is around $150 million in the US, we estimate, and double that worldwide. The test cost would be very low relative to current therapies, and so very attractive for reducing time in the ICU for these patients. And currently, we're seeking $2 million in seed round for final prototype development and pilot clinical studies to compare our test to standard renin tests. Comparables in this market include GenMark, which was acquired by Roche for $1.8 billion on a septic shock blood panel that takes an hour and a half using a bottle culture, and Inflammatix, which raised over $100 million in Series D using an mRNA test of bacterial versus viral infection that takes 30 minutes. Our management team is highly experienced. John Ashley is our CEO. He is a serial entrepreneur who's led companies through multiple FDA approvals, IPOs, and exits. I'm a co-founder and co-inventor of the patent that the company is based on, and have over 20 years of experience in biomarker development. Krish Manny, our vice president for microelectronics, has 30 years in experience in integrated circuit design, wafer fabrication, and large-scale global manufacturing and sales. We have outstanding advisors, including Daniel Bednarik, who is a leader in biotechnology and drug development at Neximmune, and Larry Busey, who is a lead investigator on the studies that led to approval of angiotensin II, as well as to determination that patients with high levels of serum renin have a benefit from taking angiotensin II, as well as Jim Nichols, who is director of point of care testing at Vanderbilt, and our advisors. So we have a very strong team, and we're excited about where we're going with this. And we hope that we could potentially hear from you. If you're interested in learning more about ResonanceDx, you can find us at www.resonancedx.com. Thanks very much for your attention.